Hello everyone, it is our honor to share with you our team's recent paper published in the journal iMeta, entitled Age-Specific Reference Intervals for Plasma Amino Acids and Their Associations with Nutrient Intake in the Chinese Pediatric Population. Dr. Yang Wen from Zhejiang Academy of Agricultural Sciences and Qing Liu from Zhejiang University of Technology are the co-authors. Dr. Wentao Liu from Zhejiang Academy of Agricultural Sciences, Associate Chief Physician Chen Weijun from Children's Hospital of Zhejiang University School of Medicine and Professor Yingping Xiao from Zhejiang Academy of Agricultural Sciences serve as the corresponding authors. With China's rapid economic development and evolving lifestyles, significant shifts have emerged in children's nutritional and health status. Plasma amino acid levels play a critical role in pediatric growth, development, and metabolic health, and deviations from normal ranges may indicate underlying health challenges during these dynamic phases. Therefore, an observational cohort study on plasma amino acid profiles in healthy Chinese children serves dual purposes, establishing essential reference data for health assessments and advancing our understanding of metabolic demands and growth, enabling early detection of disease risks and the formulation of targeted nutritional interventions. In this study, we enrolled 2009 healthy Chinese children aged 0 to 12 years as the reference cohort and 102 confirmed Phenylketonuria cases as the validation cohort. All procedures followed international and national guidelines, specifically, the CLSIC 28A3 document and China's Health Industry Standard, WS/T402-2012. First, we divided the healthy cohort into 5 age groups. Then, we analyzed plasma samples from all children to map age-specific changes in 42 amino acids and establish reference intervals. The validation cohort helped test the clinical relevance of these intervals. Additionally, we explored how nutrient intake dynamically interacts with amino acid patterns across these developmental stages. The developmental trajectories of 42 amino acids confirmed the validity of our age group design in children. Plasma amino acid metabolism in children aged 0 to 12 years showed a biphasic pattern. Cluster analysis revealed, Cluster 1, rich in essential amino acids, EAAs, increasing with age, Cluster 2, composed of non-amino acids and derivatives, decreasing with age. These clusters exhibited complementary functions to meet the demands of growth, development, and cognitive maturation. In conclusion, the 0 to 3-year critical window, marked by dramatic amino acid remodeling, is a key period for interventions targeting lifelong neurological and physical health. Age-specific amino acid profiles are vital for future health assessments. Furthermore, we validated the clinical utility of our established plasma amino acid reference intervals by applying them to confirmed phenylketonuria (PKU) cases. While phenylalanine, the core biomarker for PKU, was confirmed, relying solely on phenylalanine levels resulted in a false negative rate of 43.75%. However, our proposed phenylalanine/tyrosine ratio-based diagnostic approach, grounded in these reference intervals, reduced the false negative rate to 6.25%. This method precisely identifies metabolic imbalances caused by PAH enzyme deficiency, significantly improving early diagnostic accuracy. Such a strategy offers an optimization pathway for newborn screening programs. Given that age-specific variations in plasma amino acid concentrations align with rapid dietary transition periods during childhood, we conducted a comprehensive analysis of the correlation of nutrient intake and plasma amino acid profiles using multivariate Permanova, Spearman's correlation analysis, and multiple regression modeling. Macronutrients and micronutrients exhibited distinct age-specific intake trajectories. During the critical growth period of 0 to 1 year, fat, carbohydrates, and zinc dominated the variations in plasma amino acid changes. In contrast, protein emerged as a primary driving factor only after 2 years old. Nutrient intake was positively correlated with plasma amino acid cluster 1 and negatively correlated with cluster 2, indicating a common correlation between nutrient intake and amino acid development. Additionally, this study quantified the contributions of specific nutrients to plasma amino acid variations. In summary, this study establishes comprehensive plasma amino acid reference intervals for Chinese children and elucidates the dynamic associations between macronutrients, micronutrients, and amino acid metabolic trajectories. These findings provide critical baseline data for understanding developmental amino acid requirements, enabling early identification of risks, and guiding the design of targeted nutritional interventions tailored to pediatric growth stages.